All right, so welcome to Vera's PhD defense. She's here. You can see. Oh, that's Vera. Yeah, she's. I would say that she's every assistant professor's dream, in a way, like what you want, because she came in 2008, 19, 3 years. 3 years. She did everything that whatever I expected. She did twice as much as what I even imagined that she would do. She's very good at managing people, bringing a lot of undergrad students, working with them and helping them publish. She is actually somebody who is an enabler, doing things that I wish I'm like her, in terms of management. But research-wise, I don't want to say much because you see this is the example. She has done all. So this is Vera. Come ahead, and that's all yours.
Hi, everyone. So my name is Vera Koutnik, and today I'm going to do a PhD defense of microplastic accumulation and transport in the subsurface under weathering cycles. And first, I would like to thank my committee members for being here, Dr. Mahendra, Professor Hoke, Professor Jasby, and of course, my advisor, Professor Sanjay Mohanty. And to start, let me just go over a little bit about myself. So I graduated with a degree in chemical engineering from UC San Diego, and then I did my master's at Columbia, also in chemical engineering in 2018. And then I came to UCLA in 2019 to start my PhD in soil environmental engineering. So far, I published 7 journal articles, 4 as a lead, and then I also submitted another 2 as I currently in review, also as a lead author. I received a couple scholarships, and I also was president of EGSA in 2021, 2022. So that was a big highlight of my experience at UCLA. And now let's talk about science. The plastic pollution has been is becoming increasingly dire. The first plastics were made around 1907, and by 2016, we're already making 380 million tons per year. The production is expected only to double in the future. Therefore, the plastics are not going anywhere. They're staying with us, and so we now need to learn how to manage them and how to kind of understand what's happening to them.
Hey, Vera.
So,
Vera, this is Shelley. I don't think we're seeing your screen.
Oh.
Share the other desktop. Yeah, this is better.
Oh.
I think the slides just weren't changing.
Oh,
yeah, I think if you're on slideshows, share the other desktop.
Oh.
Not this one, the other one.
Because that doesn't work.
I see Oh, hold on. Let me.
Sorry.
Now this is faster.
Yeah, is everything good? Yeah. Okay. Hopefully. Okay. So out of all the plastics ever used, the study shows that only 30% of them are still currently in use. So majority of plastics ever made for all this many generations are actually discarded and are in landfills, open dumps, and natural environment. And this is exactly the plastics that I care about, and that's what I'm going to do in the study. How do plastics become microplastics? Due to physical weathering, like mechanical abrasion, or due to chemical weathering, like UV light, plastics naturally break down into smaller particles called microplastics or nanoplastics. Pretty much any plastic particle that's below 5 millimeters in size is considered a microplastics. And why is that important? Because throughout these years, microplastics have traveled everywhere from deepest parts of the ocean to the national parks. And so they're found everywhere in environments. And recently, they're also found everywhere in the human body. For example, a month ago, my mom from Russia sent me an article that she found there's microplastics found in human blood for the first time. In addition, microplastics are found in lungs and liver, pretty much in every part of human aspects. So it's kind of a big topic. But my research focuses on what are the hotspots of microplastic pollution. And to kind of analyze that, I did an analysis of global data, and I published 2 review articles on it. So the first one I analyzed was published in Environmental Pollution in 2021. And it looked at the distribution of microplastics in soil and freshwater environments. And it was a global analysis and framework for transport modeling. And in that study, I analyzed 196 studies from 49 countries. And I found that urban soils and urban canals were having the highest concentration of microplastics in soil and sediments. And for the freshwater, the highest concentrations were found in glacier snow and stormwater. And how does the microplastics get into the glaciers? There's no plastic, very few plastics are being used in glaciers day to day. So they're probably transported by the wind and then atmospherically deposited from the urban areas. And this microplastics transport by wind has been documented a lot. And in stormwater and urban soil, the microplastics are probably coming from the human activity that's generating plastics. Another big part of microplastics kind of cycle in the natural environment is wastewater treatment plants. So microplastics can be made by when you wash your clothes in the laundry. If you're washing your polyester clothes or anything synthetic clothing, they will actually break up and create microplastics fibers that end up getting into the gray water. The gray water goes into the wastewater treatment plants. And we actually did an analysis of 76 studies and found that the concentration inside coming into the effluent is around like 100 to thousands of pieces per liter. But concentration coming out is much smaller. So 95% of microplastics in tertiary plants are being removed from the water, from the influent into the effluent. So there's not a big problem of microplastics getting out of the wastewater treatment plant into the lakes and rivers because they're mostly removed. But when they're removed, they're not actually removed. They're being put in the sludge. And then that sludge is being deposited into the agricultural fields. So we actually find out that around 50% of all the sludge and biosolids are being land applied in the US alone, and they're used as a fertilizer in agricultural fields. So when we remove the microplastics from water, we actually reapply them and enter and put them into the sludge and then reapply them to agricultural fields. So it's kind of like a cycle of microplastics in nature where we just move them around, like humans just move them around. And what we found that due to the analysis of the methodologies used, not a lot of microplastics are being found in sludge because it's really hard to separate microplastics from really heavy media like sludge. And therefore, we actually did a mass balance and found that the land application of microplastics is much higher than expected, and that's reported now. And actually, many papers that did analysis of all the three, influent, effluent, and the sludge also found that the mass balance kind of doesn't add up. So something is happening to those microplastics. And then when they're on the soil, they actually can be moved around in urban areas by the stormwater and wind again. So by 2050, 70% of the world population will live in urban areas. And that's where people use plastics daily. So how do microplastics move around urban areas? And that's kind of the biggest kind of question that I'm trying to answer. So major carriers of microplastics in urban areas are wind and stormwater, as I showed you earlier. And that's what we found in my review papers that I published early on in my PhD. So the stormwater will then move the microplastics and then enter the stormwater biofiltration systems. And that's something that my advisor is kind of the area of the expertise. So the stormwater biofilters will then accumulate those microplastics, and then they can actually penetrate them deeper and also impact the ability of the stormwater biofilters to treat the water. And then those stormwater biofilters will then also be exposed to different weathering cycles and then kind of like, yeah. Okay, so my objective of my PhD is to identify the transport and accumulation pathways of microplastics in urban areas, specifically focusing on the urban areas and how much microplastics are there, then looking at the weathering impact and how different weather, like freeze-thaw cycles, will impact it and how they can be transported by wind. And then finally, looking more into the accumulation and the subsurface transport of those microplastics. So how deep do they penetrate and what is the risk to the groundwater? So let me talk about my research chapters. So the first two chapters were my review paper chapters, and I kind of briefly glanced over them. And then the last four are my research chapters that are research articles that I'm publishing. Number 4 is microplastics in urban playgrounds. Number 5 is urban stormwater control measures. Number 6 and 7 are both the lab studies where I investigated how deep microplastics penetrate and what affects the transport, either weathering or what characteristics of microplastics will affect it. So let me go over chapter 4. It's an elevated microplastic exposure to children in urban playgrounds, the dominant deposition pathways and population density effects. This is a paper that I just finished and we submitted it to the Science of Total Environment and it's currently in review. It's been a couple of weeks now, so hopefully we'll hear soon. So let me start by going over that in urban areas, green spaces are usually considered to be less polluted. That's where people escape to spend time and enjoy. And therefore the playgrounds, when I see them, when I walk around, I always see that they're always made of plastics. And pretty much anything that's made of plastic is probably going to shed the microplastics in their lifetime. So and in addition, those playgrounds are usually also exposed to the sun, and therefore the UV light can also break down and weather the plastics, making increasing the frequency of microplastics. And therefore, my research questions are: how much microplastics are inside the playgrounds? Where are they coming from? And what are they? To answer this question, I sampled 19 playgrounds in Los Angeles areas with a population below and above 4,000 people per square kilometer. And in red and blue, I show the locations of sampling, and also that's a difference, and I evaluated the effect of the population density on microplastics concentration. I collected sand and leaf samples and kind of quantified microplastics on them and characterized microplastics using FTIR. So I collected 3 samples on the playgrounds. I did a sample on the green star, the yellow star, and the red star. The green star shows microplastics inside the playground. The orange star shows the microplastics on the boundary, and the red star shows outside, which is around 50 to 100 meters away from the playground boundary. And I was trying to figure out if playgrounds are contamination hotspots of microplastics. So this is a picture of me actually sampling the sand from a playground. I pretty much used in all my research, I had to use everything glass or metal not to cross-contaminate my samples. I collected 3 leaf samples I collected at least 6 leaf samples, 3 inside and 3 outside, and similar for sand and soil sampling. I did composite samples at inside location, at the boundary, and outside. And then to actually extract microplastics from the sand and soil samples, I used very commonly used methods in microplastics research, which is density separation, which allows you to separate the lighter microplastics because they usually have a density of below 1.4 from the heavier soil or sand particles. And then I vacuum filtrate the supernatant. I ended up with a filter with particles on it. I dyed that filter with Nile red, which is a chemical that makes the microplastics fluorescent under UV light because it bonds to the CH bonds to it. And then I used a special device that our lab together, we collaborated with the electrical engineering department to build like a 3D printed cool little attachment to a smartphone where we just slide the filter paper in, take a picture, put the picture on a computer, and run a MATLAB algorithm to analyze a number of particles on the filter. And that allows us for a really rapid quantification method, and also allows us to quantify many samples at a time. So what we found out that urban playgrounds have an increased microplastic concentration, and the concentration decreased slowly from inside to the boundary to outside. And it was statistically significantly different between inside and outside and boundary and outside. Therefore showing that children playing inside the playgrounds are exposed to up to 5 times more microplastics than children just playing in the green areas. So when we actually analyze the microplastics, when we characterize microplastics on their chemical composition using FTIR, we found out that microplastics and sand inside the playground had a much more diverse number of polymer types. And that means that microplastics are coming from the playground structure, but they're also coming from the clothing, from the toys, pretty much from anything, the human activity. Outside was much different. It was mostly polypropylene and rayon and PET. In addition, we also analyzed, I pretty much scraped little pieces of the playground structure to analyze the origin of the microplastics, and pretty much most of the playground structures were made of PE and PP, polyethylene and polypropylene, and therefore that kind of fit together with microplastics characterization of leaf samples inside and sand samples inside. Oh. Okay. Yeah, the biggest is inside. Yeah, for my FTIR analysis, I analyzed around 40 particle pieces of sand inside the sand, and that came from 3 grams of samples. And for inside, it was actually 11 pieces because leaves have much less microplastics than the sand samples. So I think for leaves is 11 pieces for inside and outside, and for sand is I think 40 that we were able to characterize. FTIR takes a much longer time, so we have like one method to quantify microplastics and another method to characterize like a sample of that. So the take-home message of this paper is that the playgrounds are hotspots of microplastic pollution. Playground structures are made out of PE and PP, which was the dominant types of identifying microplastics. So probably some of the microplastics are coming from playground structures in addition to other things. And children playing inside the playgrounds can be exposed to up to 5 times more microplastics. So now that we know there's a lot of microplastics in urban areas, and they're coming from structures like playgrounds or any pretty much plastic structure, those microplastics can be conveyed by runoff after rain and actually infiltrate the subsurface. So to investigate that, I did another field study in Los Angeles where I looked at the microplastics retained in stormwater control measures. Where does it come from and where does it go? And Sanjay, I didn't actually know that was a song, which was kind of funny. And this paper was published in Water Research last year. So since stormwater can actually carry trash and microplastics to the bioswales and other biofiltration setups in urban areas, it's important to understand how much microplastics are coming and use other sources. For example, this is actually a picture that I took of one of the stormwater control measures in a valley, which was actually used as a trash can, which is a little sad. But also in Los Angeles, we don't have as much rain, so that kind of can explain why other factors can be in play when investigating microplastic concentration. So what we want to know is are microplastics in stormwater control measures, which I'm going to abbreviate as SCM, coming from atmospheric deposition, stormwater or stormwater, and what is the risk of groundwater contamination, aka how far down do microplastics penetrate? So to analyze that, we collected samples from 14 stormwater control measures in LA, as the map is showing, and we characterize them by different location types. Actually, we didn't find any difference, but by location type, so it's probably that doesn't have an impact on microplastic concentration. But when actually we did this, we took the samples. This is how sampling was done. We used a steel core to get the samples and then take the core at the 0.22 centimeters depth, that's the top surface, and then at 8 to 10 centimeters. And here's one of my undergrads, Joel, who helped me collect the samples. And then we analyzed, and then analyzing the data, we found that stormwater is not the only source of microplastics. So the top concentration, as expected, was much higher than the concentration at 8 to 10 centimeters. But then what was surprising when we analyzed the sample inside stormwater control measures at the inlet of the bioswales or the biofiltration systems and analyze concentration outside the SCM. So pretty much maybe like 10, 20, 30 meters away, but that doesn't have an inlet for the stormwater to flow into it. We didn't find significant difference in the concentrations. So the concentration was still high inside SCM, but it wasn't statistically significant. Therefore, there should be something else happening. In addition to stormwater coming and polluting the SCMs, it's probably atmospheric deposition. So to understand that, we also sampled the leaves outside the SCM and found that the concentration of microplastics on the leaves, which are often used as a biomonitoring system, were actually very high. They ranged from 0 to 66 pieces per centimeter squared, indicating that atmospheric deposition is a very significant source of microplastics and cannot be overlooked, especially in urban areas. And we also found that the highest concentration was at elevation of around 1.5 meters, which also means that this can increase the inhalation risk for the humans, since the people are usually, that's exactly the range where people's heads are. So now that we know there's a lot of microplastics are inside the SCM, we want to know how deep do they travel and what is it? Yeah. Oh, can't do that. I'll just do that. Oh, maybe. Oh, okay. Okay, so since now we know that a lot of microplastics are inside stormwater control measures, we want to know how deep do they penetrate. And for that, we sampled one of the locations and we sampled 5 samples and we collected the core of 22 of up to 20 centimeters. And by doing that, we were able to understand how this subsurface microplastics concentration decreases, how it changes with depths. And we actually found that the concentration was exponentially going down, indicating that there's a low potential for groundwater pollution from accumulated microplastics in the field that we sampled. So the main take-home messages is that atmospheric deposition is a significant source of microplastics accumulated in SCM, and our LEAF data showed that there's a lot of atmospheric deposition in urban areas. And finally, our analysis of one of the locations showed the subsurface concentration decreased exponentially with depths due to straining of the low pore size of the soil that's used in the biofiltration systems. So now that we know that there's a lot of microplastics in the field, in urban areas, we want to know what happens to the accumulated microplastics in biofilters and what can impact their transport. And especially if we want to check out the weathering cycles, how weathering of the environment will impact their penetration depths. And to do that, we did a study on mobility of polypropylene microplastics in stormwater biofilters under freeze-thaw cycle. And this is a paper I published in Journal of Hazardous Materials Letters last year. And this paper specifically evaluates the freeze-thaw cycles and their effect on the mobility of microplastics. So if people don't know, freeze-thaw cycle, a lot of big urban areas are located in Northern Hemisphere, and therefore they experience intermittent freezing, especially in winter. Like my hometown, I'm from Russia and Moscow, therefore there's a lot of freezing happening. So I want to know what impacts that freezing in urban areas will have on the transport of microplastics. So when freezing happens, the water actually expands. So when the water is inside the soil pores, it will expand and kind of break the soil pores and actually create different pathways for microplastics to move deeper into the subsurface. So since we know that freeze-thaw cycle has an impact on water, we want to know what impact it will have on mobilization of microplastics. And then how different media, so biofilters are usually made of different media, for example, sand or soil or biochar. So we want to know what kind of impact the different biofilter media will have on the microplastics mobility. So to do that, we did a biofilter column setup, and we actually did, we modeled biofilters and with 2 different media types. One was 100% sand, and another one was 25% sand, 25% soil and 75% sand. And that media I will call, there's a sand media and soil media, and that's how I'm going to refer to them. And then we added microplastics at top of the columns to kind of simulate the natural microplastic addition by atmosphere or by stormwater to the top of the biofilters. And we added a lot of microplastic to the top because we kind of trying to simulate the conditions over long periods of time. And then for the experimental procedure, we first pumped some synthetic stormwater into the columns. We collected the effluent to understand the background contamination of the columns, and then we start the weathering treatments. We did the freeze-thaw cycle, which was when we freeze biofilters for 6 hours at -20 degrees, and then we saw them. So we made sure that the columns are completely frozen, and then we made sure that the columns are completely thawed by the end of the experiment. So half of the columns were subject to freeze-thaw cycle, and half of the columns were subject to dry wet cycle, which was kind of a control. For dry wet cycle, we just pumped the water through them, and then we just left them drying on the lab bench. And then we repeated the water infiltration and collected effluent 40 times to kind of simulate the repeated freeze-thaw cycle that could happen in areas where something freezes at night and then it thaws during the day. So after 40 cycles, we found that freeze-thaw cycle accelerated downward mobility of microplastics in sand media more than it did in soil due to the differences in media pore size. So the sand pieces are much larger than soil. So when we have a media that's composed both of sand and soil, soil will take some of the pore spaces that used to be available for the microplastics to travel in sand-only columns. And in the left, you can see that the column where, the graph that shows depths on the y-axis and microplastic concentration normalized by the initial microplastic concentration on the x-axis. And we can see clearly that freeze-thaw cycle had a much bigger impact on the sand column, that microplastic distribution in sand columns. And that concentration decreased exponentially with depths. So, but the freeze-thaw cycle did increase the microplastics penetration. And microplastics we found even some trace amounts even at 4 centimeter depths. For the soil columns, the story was a little different, even though we did see an increased concentration of microplastics at the higher depth, at the lower depths around 1 and 2 centimeters, the microplastics were pretty much the same. The microplastics were pretty much negligible below 5 centimeters. So that shows that both the media type and the weathering treatment will both impact the microplastics transport in the urban areas and in the subsurface. And then to kind of evaluate the effect of freeze-thaw cycle more, we also check the effluent for the turbidity, just to see the difference that we can, just to see the difference in the effluent samples and how much mobilization freeze-thaw cycles actually did. And we found that both the sand and the soil columns were both impacted by that. And soil columns were actually impacted more by the freeze-thaw cycle because they release much more colloids. But then the sand samples, the sand columns, due to the bigger pore size, allowed for bigger transport of microplastics. And actually, we did SEM pictures of the effluent colloids, and we found that during dry wet cycle, the colloids in the effluent were much bigger and also less concentrated. When we analyzed the effluent for freeze-thaw cycle, we found that it was much smaller pieces, like you can see, and they were also much more concentrated. So the freeze-thaw cycle did break down the media and also got some colloids in the effluent. So the take-home messages for this paper are that freeze-thaw cycles disrupted the media and increased the downward mobility of microplastics in sand biofilters. The freeze-thaw effect was limited due to pore size of media, but the majority of microplastics remained in top 3 to 5 centimeters, and the rest followed an exponential decrease with depths. So for the areas that are not experiencing freeze-thaw cycle, the effect is not, there's no worries for groundwater pollution, but for areas that goes through freeze-thaw cycle, there's probably much bigger worry about groundwater pollution. But then to understand more what affects the downward mobility of microplastics and how different characteristics of microplastics will impact their penetration depths, we also analyzed how the specific gravity of the plastics will impact the downward mobility. Specifically, since microplastics are very versatile pollutants and they're composed from both PVC, PET, nylon, PS, so pretty much any plastic, it can be considered a microplastics when it breaks down. That means there could be a very different characteristic of those plastics, and specifically density plays a big role when microplastics are inside water. And therefore we want to know how much, what effect does that have? And to do that, we wrote a paper on microplastic transport in the subsurface by oscillating ice-water interface and the critical role of the plastic density in that procedure. We submitted this paper to Environmental Science and Technology, and it's been in review for, I guess, a couple of weeks now. So that's the last paper that I worked on in my PhD. So once you think about microplastics and plastic in general, depending on density, the density will determine the microplastic position in the water column. So if the density is high, that means that microplastics will settle. If the density is higher than water density, the microplastics will settle down. If the density is lower than water, as it will go to the top. And if the density is similar to water, they'll probably kind of stay in the middle. So as water freezes, the ice can either push the microplastics upwards or downwards or engulfs them. So which one is true and what effects that has on the microplastics transport in the subsurface? So in this paper, we are answering the question, what is the impact of freeze cycles on microplastics of different densities and the depths that they can penetrate? So we pretty much followed the exact same experimental setup for this paper. We used the bifilters. Since we found out that the standby filters allowed for bigger transport, we decided to go with that that bifilter media, and we used 20 columns, and we contaminated them with three different plastic types. Some with densities above water, which is PET, similar to water, which is PS, and below waters, which is polypropylene. And these are all one of the top 3 most popular plastics in the environment. So that's kind of makes it environmentally relevant as well. So what we found that the microplastic concentration decreased exponentially with depths, and the effect was enhanced in microplastics of high densities. So here in figure A, you can see microplastics concentration on the x-axis and the concentration and the depths going down on y-axis. And we found out that during dry wet cycles, PET, which has a density higher than PET that has a density higher than waters, actually did travel deeper into the subsurface even during dry wet cycle. But the effect for all the plastic types was more enhanced by freeze cycle. And actually for PET, we were actually able to see the PET microplastics in the effluent. Out of all the plastics analyzed and all the weathering treatment that we've done, and here we're showing 3 different plastic types and the effluent microplastic concentration, we found that there was no difference for dry wet cycle, freeze cycle for both polypropylene and polystyrene. And the difference was really apparent when we looked at the polyethylene terephthalate. And the difference was due to the heavier microplastic density. So then as a last part of that paper, we also did a force balance on microplastic particles in water. And when microplastics are in water and not close to the ice interface, they will just move according to the Stokes law, which means that the specific gravity and the size of them will determine where they move. So for PET, as shown in graph C at the bottom, they will move up trying to go to the, for PET, which is heavier than water, they will move down, and for polypropylene and polystyrene, which are lighter than water, they'll go up. And then but then when situation is closer to the water, closer to the ice, situation changes a little bit, because when the ice interface is moving and there's a moving ice front going down, not going down, just moving the water, that means that when the particle is close to the ice, if the particle is small, the density effect is not as apparent, because if the particle is so small, the ice interface will actually have a bigger impact on it. But when the particle is bigger, both the gravity will play a role, but also the moving ice interface. And that will mean that the denser microplastics like PET will have a higher velocity when they're close to the ice front, when they're bigger and close to the ice front, therefore increasing their mobility in the water and the subsurface. So the take-home messages are the freeze cycles could disproportionately accelerate the downward mobility of dense microplastics. Also, when the microplastics are small, they could be pushed by high velocity by ice-water interface, but the density does not play such a big role. And the PET and PVC, and this is the 2 types of plastics that are really commonly found in the environment. So PET is all your water bottles are made of that. PVC are pretty much the pipes and things like that. And those are the 2 microplastic types that are much heavier than water. They have a much more increased risk of groundwater pollution, especially since we only did around 35 to 40 cycles for this paper and the last paper. Therefore, when in the areas which experience much more freezing and thawing, like for example, if the area experiences freezing and thawing every single day, that means the place will have at least like 90 freeze cycles per year. And so annually, that can actually play a much bigger role. Therefore, future studies can look and actually study the impact of the increased freeze cycles and the PET and PVC. So I'll just quickly go about talk about main findings for each one of my research chapters. In chapter 2, that was my global distribution of microplastics. And we found that global microplastic concentration varies over 8 orders of magnitude, and urban areas and glaciers are the hotspot. In chapter 3, I discovered that there's a lot of trillions of microplastics pieces are released annually via wastewater biosolid applications on land in US alone, therefore reintroducing microplastic into the natural environment. In chapter 4, I found that children playing inside in playgrounds are exposed to up to 5 times more microplastics than children playing outside the playground boundary. In chapter 5, I found out that stormwater biofilters accumulate microplastics and deposited by air and the stormwater, but the groundwater pollution risk is pretty low. And then in chapter 6, I found out that freezer cycle increased the microplastics penetration depths, and the effect was more prominent in biofilters with higher sand content and bigger pore size. And in chapter 7, I showed that the freezer cycle disproportionately accelerated downward mobility of denser microplastics, and therefore future studies should look at that. Okay, and for future studies, just generally in microplastics research, we need to come up with better methodologies to characterize smaller microplastic pieces, because the bigger pieces are being strained out by the pore size of the soil, but nanoplastics can still travel through the through the pores and penetrate into groundwater. In urban areas, we need more studies to investigate a suspension of mechanism of microplastics into the air, which is something hopefully we're going to do in our lab. And in stormwater biofilters, we want to analyze the effect of microplastic deposition on biochemicals processes in the subsurface, since stormwater biofilters are usually serve a function to treat the pollutants, and therefore biologically treat the pollutants, and therefore the increased microplastic concentration can have an impact on that. So we kind of want to know what impact is that. And now to the acknowledgements. First of all, I want to say a huge thank you to my undergraduate researchers. I would not be here without them. They helped me day and night in lab, and I don't know, I'm just really proud of every single person. They helped me publish papers, they helped me do all my experiments, and I would never be done in 3 years without them. Sarah, Francesca, Joelle, Jocelyn, Albert, Anna, Leah, and Michelle, thank you so much for helping me out. And for the acknowledgements, first, thank you so much to my committee members, Dr. Sanjay Mohanty, Dr. Shelly Mahendra, Dr. Eric Hogue, Dr. Dara Jasby for being here, supporting me, and giving me your time. And thanks to my amazing lab mates, Jamie Leonard and Dr. Anesh Borsakor. And of course, everyone at C-Lab for helping me out and supporting me. And of course, thank you to my collaborators, Professor Hatis Koydemir, Dr. Derek Sang, Professor Aydogan Özkan for designing our microplastic identification 3D printed little smartphone attachment, Professor Sujit Ravi for all the particle size analysis and for help with writing, and Dr. Vin Kauger, who is now at Moore's Institute for Plastic Pollution Research, for providing and teaching me how to use FTIR. And also I would like to say thanks to UCLA's Samueli School of Engineering for providing me with funding and support and resources and lab space, Civil Environmental Engineering Department. And of course, I also received NSF Infuse funding through my last year to help me support my studies as well. Thank you so much. And now my next steps, so I got a job at Boston Consulting Group. So I'll be starting in their Los Angeles office in August 1st. So I'll be around here in LA, but I'm really excited for my job. And thank you so much.
Wow, catch a breath.
Yeah, this is excellent.